everyone, welcome back to my do an assignment because it's from works, and the assignment needs to be a YouTube video. Today we will be reviewing Sarcoplasmic Hypertrophy Program 5.0 Part 1 by Genetic Beast. The capitalization is important. First off, there's 5 main points that I want to touch on, though they all flow into each other. First off, he says that sarcoplasmic hypertrophy is for people who don't have time for hyperplasia. But we aren't going to touch on that. Instead, our actual first point occurs when Mr. Genetic Beast, now shortened to Mr. JB, talks about glycogen loading. We train the muscle to load glycogen into the muscle so that the cells around the muscle swell, and over that time, they become real. Let's talk about where glycogen is stored. A study by Frederick Seger and Ekblom in 1989 explored the idea and found that glycogen is stored in 5 different places: subsarcolemmal space, intermyofibril spaces, and different intramyofibular spaces. Now notice how none of these places are actually cells themselves, but instead areas within or around cells. Mr. Genetic Beast most likely was misinformed and assumed that sarcoplasm by itself was an individual cell. Unfortunately, since they aren't, it's unlikely that these cells would ever become real. However, he is correct in stating that glycogen is stored with water, which does increase the water weight of said muscle and makes it look bigger, as the sarcolemma increases in size. Mr. JB then talks about why sarcoplasmic hypertrophy works, and his example is that because glycogen loading increases the amount of water in the muscle just like creatine, then it just works. Because creatine does build water in the muscle and makes it bigger, and that's why people become stronger. Well, creatine does increase the cross-sectional area of muscle fiber types and retains water. Volk et al. Also demonstrates that it also allows people to induce a greater training stimulus to allow for greater adaptations. It also increases the rate of photocreatine resynthesis as shown in Greenhouse et al., which lends itself to a greater recovery rate. So while it does retain water, creatine also has multiple other benefits. Next, he talks about his optimal go-to food for sarcoplasmic hypertrophy: potatoes. But why potatoes? Though he mentions that they contain complex carbs and more importantly, starch, there is an additional reason. A study by Burke, Collier, and Hargreaves looked at the effects of glycemic index of both exercise carbohydrate intake on muscle glycogen storage. They found that compared to eating foods with a low glycemic index, eating foods with a high glycemic index increased the muscle glycogen content after 24 hours of recovery after a glycogen depleting bout. And luckily for Mr. JB, according to the international tables of glycemic index, most potatoes are categorized as high glycemic index foods. Speaking about glycogen depleting bouts, Mr. JB talks about aiming for 5 sets of 8 to 10 reps above 50% 1 RM to stimulate sarcoplasmic hypertrophy. Though sarcoplasmic hypertrophy itself cannot be disproven, that doesn't change the fact that it's important to deplete glycogen, and you plan on focusing on glycogen loading. Jennison et al. Shows that glycogen content is a strong regulator of glycogen synthase. By increasing glycogen content in the muscle, you decrease glycogen synthase activity, as the glycogen phosphorylates the enzyme, making it less active. Therefore, if you don't deplete the glycogen in the muscle in the first place, you'll be making minimal amounts of glycogen, no matter how many potatoes you eat. Finally, another regulator of glycogen synthase is insulin. It promotes glucose in uptake and increases glucose 6-phosphate in the muscle, which stimulates glycogen synthase to make more glycogen. This can be shown in a study done by Buska et al., where they incubated isolated soleus mice muscle with and without insulin. As you can see, in the state with insulin, both glycogen synthesis and glycogen content are significantly increased when compared to without insulin. Mr. JB mentioned how you would want to promote insulin resistance in order to help with sarcoplasmic hypertrophy, but it's the opposite. Insulin resistance implies that our cells are less sensitive to the hormone, and therefore the stimulating of glycogen synthase is inhibited. Overall, though Mr. Genetic Beast makes some good points about glycogen loading, the reasoning behind the mechanisms are a bit wrong. Nonetheless, that doesn't mean that he can't learn from this and dive into research in order to understand muscle hypertrophy on a deeper level and help clarify his statements in future videos.